So this is our last lecture on this topic, replication, virus replication. So in the previous sections we discussed about the normal replication. Today we'll discuss about the defective replication, defective unproductive replication. This is defective, and what we studied was productive replication. What was productive replication? Yes, attachment, then penetration, then uncoating, then transcription, translation, replication, all steps, again assembly, maturation, and release of the virus. This was the mature or infectious virus. For example, here 10 virus enters and 100 comes out. 10 virus they enter, 100 comes out. This was the productive replication. What is defective replication? Similarly, all the steps are same. Suppose 10 enters, again attachment, penetration, uncoating, transcription, replication, translation, assembly, maturation, and release. So in defective replication, what is different with the productive one is, here 10 enters and here also 10 enters. But in this case, these all hundreds, they are not infectious. They are not out of, if 100 are coming out, they comes in this ratio. For example, 80 viruses they are defective and 20 they are infectious. Defectives are non-infectious. So this is how a productive replication ends with 100 mature infectious viruses, and a defective replication ends with, out of 100, 80 are defective and 20 are infectious. How this defective process takes place, we'll discuss with one example. In 1954, there was a scientist, Peter von Magnus. Peter von Magnus, he was working with embryonated eggs. He was working with embryonated eggs, and which root? Allantoic root. Allantoic cell root he was working. What he did, he took viruses, high dose, or we can say high dose, or we can say high multiplication of infection. High dose or high multiplication of infection, MOI. With this high dose embryonated egg allantoic root, which virus was studied? Influenza virus. So this, when von Magnus, he studied with the influenza viruses. He took high dose, and what he performed? He performed multiple multiple passage. Multiple passage in the embryonated egg. What he found that this virus, it enters, and what comes out, normal infectious and defective infect interfering particle, and this normal is known as helper virus. Why it is called helper virus? Because in the successive passages after this, if again we pass it through the cells, means different passage, then both helper and DIP, defective particles, they will enter and they will utilize the resources of the cell, and more DI particle and less helper or normal virus comes out. So this was called as drop in yield. We'll discuss these two steps in detail, but in short, what Peter von Magnus did, he took high dose of influenza virus and he took embryonated eggs, and then passages multiple times he passed these viruses, and what he found that not only the normal infectious viral particle comes out, along with some defective particle also come out. When these virus particle again they infected the cells, then these two, one is helper, one is normal, one is defective. Defective virus lack some machineries, lack some genome, and it take the help of this normal virus, and both utilizes resources, and the end result is more defective particle, less helper or infectious particle, and this is known as drop in yield of the infectious particle. So what happened at this place, why helper are formed, why DIP are formed, we will discuss now. This is the cell and this is the influenza virus particle. Again it enters, penetrate, then it releases all the 8 segments. It releases all the 8 segments. It releases all the 8 segments. And these segments are further taken care by which enzyme? RDRP. RNA dependent RNA polymerase. The bad about this enzyme is it is error prone enzyme. Why it is error prone? Because it lack proofreading activities. Proofreading activity. Because of this, there is more risk of mistake. More risk of mistakes during replication process or transcription process. For example, these are 4 progeny viruses which are coming out of the cell. In this virus, segment number 4 is replicated more number of time, and this 4 encodes hemagglutinin. So HA will form more than NA. So this is the virus particle coming out with this defect. More surface glycoprotein HA hemagglutinin. What happened with this one? Segment number 6, which encode NA. It is replicated multiple times. So here Na is more than hemagglutinin. So both are defective one. This is having more HA. It means it is more infectious. It is more infectious because it help in attachment with the host cell, and this is less infectious. This is less infectious. This virus is having some loss of some gene particles. These are the segments which are deleted. Some segments are deleted. So, for example, number 4, 5, 6, these segments are deleted. So, this is also defective interfering particle, and this is the normal happy particle. This is normal, and normal is also known as helper. This is helper for all these three. So why these different defective particles are formed? Because of some defect at the step of replication. Some defect may occur at the place of assembly, maturation, all the steps. So these are the defective one, and this is the normal one. One reason is the RDRP, which is error prone because it lack proofreading activity. One more reason could be RNA genome is unstable. RNA genome is itself unstable. And one more reason is the number of segments. 8 segment. So these are the few reasons why defective particles are formed in excess as compared to normal. So when this is the case, suppose first time this was the cell and this is normal, and these three are defective. We call them defective particles. In the next passage, when they enter one more cell, this normal also enter, and these three defective, they also enter. Suppose this is the case. In this case, normal releases its 8 segment, and these viruses also releases their genome after uncoating. But in this cell, resources are limited. What are resources? Polymerase enzyme, nucleotides, and other resources, ATP and all. So these resources are utilized by this normal as well as the defective one. So these enzymes, for example, RDRP, if these segments are deleted and small, it will replicate faster. small segment faster. So this enzyme is busy with defective particles replication, assembly, maturation and release. So these are defective. So what they are taking help from this because they lack some segments or some enzymes and other resources. So they are taking, these defectives are taking help. Not only they are taking help, they are also number two, they are interfering. They are interfering with the replication of normal. So they are taking help of this. So this is known as helper virus, which is the normal one, infectious one. And these, they are interfering particles. They are interfering particles and also they are defective. So we call them DIP. First of all, they are defective, then they are interfering with the normal replication. So defective interfering particles. And the normal one is the helper. This virus is helping these by providing some segments for their replication, for their assembly, for packaging and all. So at molecular level, this occur with the influenza viruses. We'll take one more example, VSV, vesicular stomatitis virus, which belong to rabies family. Rabies family, that is Rhabdoviridae, and rabies family means it is having bullet shape. It is having bullet shape. So this is the bullet which enters and it is having a single RNA and it releases uncoating and release virus. What happened with VSV virus? In this case, this 2/3 portion and this is 1/3. This 2/3 is deleted. This 2/3 portion is deleted. So what is the result of this? For example, some viruses formed, they are normal. They are normal bullet. They are normal. So we call them B particle. They are called B particle. And those particles where 2/3 portion is deleted or truncated, they are known as T particle. They are T particle. In this, only this 1/3 part is present as a genome. So both have same morphology as a bullet shape, but this is a small bullet. This is small bullet. So B particle are normal, T particle are DIP, defective interfering particle. They have 2/3 part is truncated, deleted, only 1/3 is remaining. And because of this small size, they are small bullet shape. So both have same morphology. One is B particle, one is T particle. So their genome is shorter. So in this way, vesiculostomatitis virus, normal B particle and these particles are formed. Again, if this B particle and this T infect the same cell, then B will provide help to the T particle and they will multiply. So T will come more and B will come less. So this is the result of next passage or next application. So Peter von Magnus performed his experiment that was in laboratory, that was in embryonated egg. But what happens in natural infection? What happens in vivo, not in vitro? What happens inside a normal cell? This is the normal cell, and this is infected by IMDH. So there are some viruses which shows defective replication. I means influenza, M for measles, D for dengue, and this is hepatitis A and C. So these viruses, they infect the cell naturally. Then what happens? DIP form. This DIP may alter pathogenesis of disease. Pathogenesis of the disease. How this DIP they alter the pathogenesis of the disease? Number one, they interfere with replication of H helper virus. They interfere the replication of helper. Helper is having full length genome and DIP is having shorter genome. So the enzyme RDRP will replicate the shorter genome faster. Replicate faster. It means DIP and helper both are competing for the same resources. Both are competing for the same resources. What is the end result of this? Thus less helper will come out and more DIP. So less helper means less infectious will come out. Less infectious comes out is known as reduced viral load or milder disease. So the pathogenesis of disease from the severe disease, it becomes milder. Why it becomes milder? Because of formation of this DIP. Because they reduce the number of infectious viral load. So disease become less severe or mild form. So this is the one way by competing with the main infectious virus, these DIP reduce the severity of the disease or they alter the pathogenesis of the disease. Second way is number 2, they activate or stimulate innate immunity, innate immune response. These DIP stimulate or activate the innate immunity. How they stimulate? They stimulate production of number one interferon, type 1 interferon, which are nothing but alpha beta. Interferon are produced. Where these are produced? These are the nearby cells. These are the nearby cells. The DIP comes from this normal cell. They will stimulate these nearby cells, and what comes out? Interferon type 1. Number 2, they stimulate the production of pro-inflammatory cytokines. For example, interleukin 1, 6 and tumor necrotic factor TNF beta. Not beta, that is TNF alpha. So these are the cytokines. These cytokines, they will recruit inflammatory cells. They will recruit the inflammatory cell to fight with infection. So this is the natural infection of these viruses with the cell. What comes out? DIP. DIP will alter the pathogenesis of a disease. First way, they interfere with replication with helper or normal virus. In this way, they reduce the number of infectious particles and reduce the severity of disease become mild. Number 2, they activate innate immunity. Stimulate the production of interferon from the nearby cells. Number 2, pro-inflammatory cytokines, which recruit inflammatory cells to fight the infection. So we can say DIP acts as natural antiviral agent. DIP may act as natural antiviral agent because they alter the pathogenesis of disease. They interfere with replication, and the end result was mild disease. And here end result was host defense increases because of interferon cytokine production. So in this way, mild disease, whole defense activation, they act as a natural antiviral agent. And these DIP may have some therapeutic potential. We can use them as a prevention of some virus infection, or we can use them treatment of some viral infection. So they have therapeutic potential as antiviral agent. So we can also call them novel antiviral agent. So this is the clinical utility of DIP particles. So DIP formation in DNA viruses and DIP formation in RNA viruses. This is less frequent. This is less frequent. And in case of RNA, it is most common. It is most common. Why it is less frequent? Because they use DNA polymerase enzyme. DNA polymerase is having proofreading activity. So because of this proofreading activities, there are less error or less risk of mistake. RNA viruses most common because they uses enzyme RDRP, and this RDRP is having lack proofreading and more prone to errors. So DIP formation is most common in RNA viruses because of this reason. So which DNA viruses? They show there are three example. Number one, adenovirus. Number two, herpes. Number three, pox. So these three viruses, under certain condition like high dose, high multiplication of infection, they may show DIP formation. And in case of RNA viruses, it is most common. It is most common. So after this DIP formation, the next topic with the replication is formation of inclusion bodies. Formation of next topic is inclusion bodies. So inclusion bodies represent site of replication. What they represent? Site of replications. Site of replication. So inclusion bodies may have size, shape, staining properties, and they have different location, like inside the cell or nucleus, different location. So number one, intracytoplasmic, then intranuclear, and both intracytoplasmic and intranuclear inclusion bodies. Intracytoplasmic, number one, Negri bodies, in that is rabies. Number two, Guarneri bodies in smallpox, that is variola. Not variola, it is Guarneri bodies, smallpox only. Number three, Pascan bodies, that is seen in variola viruses. Number four, Foulpox, that is Bollinger bodies. Bollinger bodies in Foulpox. And number five is Botryoid bodies. Number five is Botryoid bodies. They are found in porcine circovirus 2. Porcine circovirus. So these are the intracellular inclusions. Intranuclears may be of two types. You can remember them as a dry cow. Dry cow. There are two cows, dry cow A and dry cow B. So human in medical they use this Cowdery type A, Cowdery's type B. So you can remember with tricow A, tricow B. These inclusion bodies they are large, they are large, but they are few and large, and they are granular. They are granular. And Cowdery type B, they are multiple. They are multiple. They are small and they are circular. Some books write it circumscribed. Circumscribed. So these are the type A, type B. Few in number, large in size, granular, multiple, small, circular. What are the example? Example is herpes and yellow fever. Herpes and yellow fever. These yellow fever are known as Torres bodies. They are known as Torres bodies. And then these type B examples are 1, 2 and 3. Number 1 is adenoviruses. First we'll take poliovirus, then then cytomegalovirus, which is having owl's eye appearance. So there are 3 examples of Cowdery type B: polio, adeno and CMV. Poliovirus inclusion bodies are acidophilic. They are acidophilic. And these two, adeno and cytomegalovirus, these both are basophilic. Basophilic, blue in color. Then both intracytoplasmic, intranuclear, they are paramyxoviruses, that is canine distemper and measles. The measles inclusion bodies are known as Warthin, Finkeldy bodies. Warthin-Finkeldy body. Measles inclusion bodies name. So these are 3 types of inclusion bodies. One is cytoplasmic, nuclear and both. Intranuclear has 2 types, Cowdery type A, B, and these are the example. Then fowl pox. In case of fowl pox, this is the cell, and this is the inclusion body. And inside this is known as Borel body. And the blue color, this outer one is the Bollinger body. This one is the Bollinger body. So this is intracytoplasmic. Bollinger body is large. It is round. It is large and round, and it is acidophilic. This one, Bollinger round body. Inside this Bollinger body, we have Borel body. What Borel body represent? What they represent? They represent actual viral particles. They represent actual viral particles. These viral bodies are also known as elementary bodies. Elementary bodies. These are viral body or elementary body. They are present inside the Bollinger bodies. So they represent actual viral particle, and in nature they are basophilic. They are basophilic. So main Bollinger body is acidophilic and Borel is the basophilic. This is large round and these are representing actual viral particle, and they are small and they are granular. They are small and granular. So these are the features of Borel body: small, granular, elementary, actual viral particle, or they are basophilic. So that is present in fowl pox. Next topic is cytopathic effect. When a virus multiplies inside a cell, what is the effect produced? That is cytopathic effect. The CPE, cytopathic effect, is morphological and structural changes. Number one, paramyxo. In case of paramyxo, there is formation of syncytium, that is giant cell with multiple nuclei. And it is having faint basophilic intracytoplasmic inclusion bodies. Intracytoplasmic. Number two is your pox viruses. This cell swelling. Cell swelling is the cytopathic effect. Number three is the herpes virus. This is stranding of the cytoplasm. This cytoplasm becomes like a strand. If this is the cell, then cytoplasm will go out like a strand. These are known as strands of cytoplasm. Number four is adenoviruses. Here, cells become enlarged, rounding, and like a grapes. So, grapes-like appearance. Number five is the foot and mouth disease virus. Foot FMD, that is ballooning degeneration. That leads to formation of the vesicles. So these are the morphological changes. Somewhere cells become swollen, cells become multinucleated, giant cells become ballooning, cell rounding, grapes-like. These are the cytopathic morphological changes because of replication. So here we end our topic, replication.